All right, welcome to our explainer. Today, we're diving deep into the code of life itself. You see, we've hit this incredible point in genomics, where we can generate way more biological data than any single human or even a team of humans can possibly understand. Now, this isn't just some technical hurdle, it's what we're calling a crisis in perception. So let's get right into it. Take a look at this number, 150 gigabytes. That is the amount of raw data that gets spat out from sequencing just one single human genome. To put that in perspective, imagine trying to find a single typo in a library that has millions of books, and each book is thousands of pages long. That's the sheer scale of the challenge we're up against. And this explosion of data, it really is a double-edged sword. This quote from our source material, Applications of Deep Learning in Genomics, nails it. On the one hand, we've got this unbelievable opportunity to decode the secrets of disease, of life itself. But on the other hand, we have the monumental challenge of just making sense of all of it. Which brings us to our first section. You've got to think of this as a literal flood. The amount of biological information coming from our modern sequencing tech, it's a torrent that has completely overwhelmed the traditional ways we used to analyze our own biology. And this is the absolute key. Human perception, our ability to see and understand, has become the bottleneck. We've built these incredible machines that can read the book of life faster than any person ever could, but we can't keep up. And when that happens, the power to see the patterns, to interpret the meaning, well, that power shifts away from us and toward the systems that can actually do the math. So let's get a little more specific. What does the state of flood actually break? I mean, what are the specific parts of the process where the old methods, the ones that kind of relied on a human being in the loop, just can't hang anymore? Okay, take a process called DNA fragment assembly. Our sequencing machines, they don't read the whole 3 billion letter genome in one go. Nope, they chop it up and read millions of tiny overlapping pieces. Putting it back together it's like trying to reassemble a billion shredded copies of the same encyclopedia, and just to make it fun, some of those copies have typos. It's a puzzle of almost unimaginable complexity. And this is exactly where the old system just hits a brick wall. Our source material puts it bluntly, saying that the complexity renders traditional assembly strategies, and I quote, useless in many situations. It's not that the old tools were bad, not at all. It's that the game itself has changed so completely that we need a new kind of tool. It's not just helpful anymore. It's absolutely necessary. So what is that new tool? Well, it's a new kind of lens for seeing patterns in all that noise, and it's called deep learning. It's a branch of artificial intelligence that just so happens to be exceptionally good at finding meaningful insights inside these massive, complicated, and frankly messy datasets, which is a perfect description of the data genomics is producing right now. You know, this really shows what a huge shift it is. Traditional analysis, it looks for simple A causes B relationships and requires a human scientist to tell it what features to even look for. But biology is messy, it's full of these complex, indirect connections. That's what nonlinear really means. Deep learning loves this complexity. Instead of being told the rules, it figures out the rules for itself, straight from the data, finding patterns on a scale we just couldn't see before. And deep learning isn't just one thing. Think of it as a whole new toolkit. You've got different tools for different jobs. Convolutional neural networks, or CNNs, they're great at spotting images, which means they can spot specific patterns or motifs in a gene sequence. Then you have recurrent neural networks, RNNs, which are designed to understand sequences, making them perfect for looking at how genes are expressed over time. And transformers? Well, they're incredibly powerful at seeing the whole big picture, how different parts of the genome, even ones that are really far apart, can influence each other. And that right there, that gets to the heart of it. A really good way to think about deep learning in this context is as a super powerful signal amplifier. Imagine you're in a really noisy lab. Deep learning is the thing that can pick up on those faint, complex biological signals that were just getting drowned out by all the static before. So you've got this incredibly powerful new lens. What can you actually do with it? This is where the story gets really, really exciting. We start to unlock a future of medicine that is so much more precise and personalized than anything we've ever seen before. I mean, we are talking about being able to predict the function of totally unknown genes, to pinpoint the tiniest genetic variations that lead to disease, and even to develop treatments that are tailored to your unique genetic makeup. This technology can automate huge chunks of biological interpretation and just dramatically speed up the discovery of new drugs. This is the promise. But there's always a but, isn't there? And this is a big one. This powerful new lens, it isn't made of glass. It's made of millions and millions of mathematical calculations inside a neural network. And that leads us to a really critical, almost philosophical question for the future of science and medicine. And this is the crisis at the very heart of this explainer, the crisis of trust. For all of its amazing power, this shift brings with it a whole set of darker, more complicated challenges. And they all center on one question. Can we, or, should we, trust an answer if we don't understand how it got there? This is famously known as the black box problem. The source material points out that this opacity, this lack of clarity, is a huge barrier to actually using this in hospitals. Doctors, patients, regulators, we all need to have faith in the predictions a model makes. And that's pretty hard to do when its internal logic is a complete mystery. We get the right answer, maybe, but we have no idea why it's the right answer. Just look at this diagram for a second. I'm not even going to try to explain what it is because, in a way, its sheer complexity is the explanation. This is the black box visualized. The path from an input on one side to an output on the other goes through a web so tangled that trying to trace the reasoning is practically impossible for a human brain to follow. And the black box is really just one piece of a much larger puzzle of risks. I mean, what if the genetic data we train these models on isn't diverse enough? Then the models will be biased. There are huge privacy issues with our most sensitive biological information, and the cost to run these things is so high that this power could end up concentrated in the hands of just a few big players. So, with all these risks, you have to ask, why is this system becoming so dominant so quickly? And that brings us to our final point. Because, despite all its challenges, this is a system that is incredibly difficult to slow down, let alone rewind. And here's the simple, powerful reason why. The whole system is self-reinforcing. Deep learning comes along and solves the too much data problem. It gives us the ability to handle what was completely unmanageable before. And once our institutions, universities, hospitals, drug companies, once they build their workflows around that new capacity, well, going backward just isn't an option anymore. This reinforcing loop is what makes the system so incredibly powerful. It all starts with more genetic sequencing, which, of course, creates more data. That data is then used to train bigger and better deep learning models. Those models then uncover new biological insights, which proves how valuable they are. And that value, in turn, drives more investment right back into, you guessed it, more sequencing. The cycle just keeps getting stronger and faster. So the most important takeaway here is that we can't just look at this as the next step in technological progress. It's much deeper than that. This is a fundamental shift in the very nature of scientific authority. It's a change in how knowledge is made, how it's validated, and ultimately how it's trusted in medicine and in biology. And that leaves us with this final hanging question, which is really the central dilemma of this new era. We have built tools that can perceive patterns in our own biology that are literally invisible to us. And the real crisis of perception is figuring out what it means to trust those tools, and maybe more importantly, who we trust to wield them.